Welcome. In the last session, we studied about actin filament and myosin filament, and also we have seen about the triad system. In this session, I would like to discuss about mechanism of muscle contraction. So we shall see about mechanism of muscle contraction. So this mechanism of muscle contraction is explained by sliding filament theory. Is explained by sliding filament theory. And this sliding filament theory was proposed by two persons, named Gene Hansen is one person, and another person, Andrew H. Andrew F. Huxley. So they proposed this in 1954. Proposed in 1954. So now let us see how this sliding filament theory, uh, okay, and what how it explains the mechanism of muscle contraction. So here, before we go into it, a few more points that we need to recollect that on the actin filament, we know there are okay, so two F actins and two tropomyosin, and on that tropomyosin we have seen at regular intervals there is a distribution of, okay, troponin. So you can see this is how the troponin is present, and this one is tropomyosin, and this is troponin. And we have seen that this troponin has three subunits, and those subunits are TNT, okay, and then TNI and TNC. So in short, I would like to tell that this TNT is the one that has strong affinity for tropomyosin. Strong affinity for tropomyosin. Then TNI, this one has the strong affinity for actin. Has strong affinity for actin. And the TNC has strong affinity for calcium ions. Okay, right. So this is the thing which we have to keep in mind. So very important. So about these troponin molecules here. Okay, so here these troponin molecules are intermittently attached along the sides of tropomyosin. Okay, so this troponin is intermittently attached along the sides of tropomyosin. Okay, right. Next, here in the resting state, so means when the muscle is not contracting. So in the resting state, what happens is this tropomyosin molecules will lie on the top of active sites on the actin. So you may see here, okay, this is an actin. So G actins. So you can see here this tropomyosin molecule will cover the active site on these actin molecules. So these are all the G actins of actin filament. Actins of actin filament. Now this tropomyosin in the resting state. So in resting state, so when this muscle, there is no muscle contraction, the tropomyosin molecules will lie on the top of active sites, will lie on the top of active sites of the actin strands of the actin strands. Actually we have here, on case of the actin filament, so we find active sites. So if this tropomyosin slightly slides out, so then you can see there will be active sites on this actin filament. So here I'm just showing that. So this is the actin filament, and so just the tropomyosin just moved away, so that now it shows the presence of the active site. So here are the active sites on these G actins of actin filament. So these are active sites. It is to these sites the myosin head will come and bind, and that actually initiates the process of muscle contraction. That we'll see in case of this sliding filament theory. Then here one more point. So when this tropomyosin covers the active sites, there will be no attraction between the myosin head and the actin filament. It is the myosin head and the actin filament that will come against one another and attach together, and that results in initiation of muscle contraction. So we'll see the sliding filament theory now. The contraction of muscle is initiated when a signal from the central nervous system comes through the motor neuron. So here we will see the motor neuron. So that have the axon terminals, and those axon terminals will have the synaptic knobs that they will come in contact with the the muscle fiber. So this is the neuron. So we'll see this synaptic knob will be resting on the plasma membrane of the muscle cell. So how so this is that I will just in a moment I will tell you. So here you can see this is the sarcolemma, and so this is the synaptic knob, and the motor neuron. So this is a motor neuron, and this motor neuron brings the information from the central nervous system, and finally it reaches to the synaptic vesicle, and then this information will enter into the the muscle fiber. So this is the muscle fiber and helps in contraction of those sarcomeres that together result in contraction of the muscles. So before we go, we need to even see about something called the neuromuscular junction. So this is the point where, okay, where the synaptic knob of a motor neuron makes the contact with the sarcolemma, is what I call the neuromuscular junction. It's called neuromuscular junction. Okay, and this neuromuscular junction is also called motor end plate. Is called motor end plate. Now I will show you a three-dimensional picture of how this neuromuscular junction is present. So here I am showing you the picture where, okay, the motor neuron forms, okay, the synapse with the right muscle fibers. So here are the muscle fibers. So these are the muscle fibers. So each one, so this is one muscle fiber, this is another muscle fiber, and we know all these muscle fibers together form, okay, the fascicle. Here you can see so many. So already we have seen, okay, the transverse section of this muscle, and inside you can see all these red dots. So those red dots are the myofibrils. So they are myofibrils, and you can see here the motor neuron. So it branched, and then it shows, okay, lot many synaptic knobs that are in contact with the sarcolemma of these muscle fibers. Okay, so the point where it comes in contact, we'll see. Okay, so this is the synaptic knob, and so this is the sarcolemma. So it shows, so somewhat like this. Okay, so this is the sarcolemma of muscle fiber, and this is the synaptic knob from the motor neuron. And here inside this we find a lot many, okay, vesicles. So these are the vesicles that consist of neurotransmitters, and along with that you may find a lot many mitochondria. So you will find uh, mitochondria. So okay, and here also you find, in the muscle fiber also you will find so many mitochondria. So these are all the mitochondria, and these are the synaptic vesicles, the synaptic vesicles, and these synaptic vesicles will contain neurotransmitter, okay, like acetylcholine. So synaptic vesicles will contain neurotransmitter, and that is acetylcholine. So here when there is an impulse, so it results in the release of this, okay, synaptic, I mean acetylcholine from the synaptic vesicles into this, okay, synaptic cleft. So you can see here that this small gap is called the synaptic cleft. Okay, so this is what we call the neuromuscular junction. So here all whatever here showing, so this is a neuromuscular junction, is a neuromuscular junction, also called motor end plate. It's called motor end plate. So here again I'm showing one more diagram. So you can see here this is the axon of the motor neuron. So this is the axon of the motor neuron. Okay, and this is the synaptic terminal. So this is the synaptic terminal, and it's forming the neuromuscular junction or motor end plate, and this is how it looks at that point. So this is what I mentioned, the neuromuscular junction, so which is also called the motor end plate. It's called motor end plate, and you can see here there is a small point entering. Okay, so these are okay, what we call that enters into the T tubule. So you can see small depressions on this the sarcolemma. So this is the sarcolemma. Okay, so already they have mentioned here the plasma membrane of muscle fiber, this yellow color one, and inside that you can see this one structure is the myofibril. The one structure is the myofibril, and this is the T tubule. So yesterday I was talking about the triad system. Okay, so this T tubule, and there is a blue color, okay, which is present on either side of the T tubule, what I call it sarcoplasmic reticulum. That is the endoplasmic reticulum present, okay, on either side. So this sarcoplasmic reticulum present on this side and this side of this T tubule, together it will constitute the triad system. Okay, so this is how the axon of the motor neuron forms the synaptic it forms the neuromuscular junction here. So this motor neuron will bring the impulse, okay, into this synaptic knob, and once it comes into the synaptic knobs, okay, the synaptic vesicles will finally come and bind, okay, with the the membrane of this the synaptic knob, and then it releases the neurotransmitter into the synaptic cleft. So you can see the neurotransmitter is released. So this is the acetylcholine, which is released into the cleft, and that binds with the receptors that will be present, okay, on these membranes, and that results in undergoing the depolarization, and the okay information will reach into the sarcoplasm. Okay, and finally the information reaches to this endoplasmic reticulum. Once the information reaches to the endoplasmic reticulum, we know the endoplasmic reticulum is storehouse of calcium ions. So these calcium ions will be released from the endoplasmic reticulum. Okay, so the calcium ions released from endoplasmic reticulum. Okay, so here the point is that when the neural signal reaches, okay, the junction, that is the neuromuscular junction, what happens from the synaptic knob, the neurotransmitter called acetylcholine will be released. Okay, and this okay results in the generation of the action potentials in the muscle, and that spreads through the muscle, and it finally reaches to the sarcoplasmic reticulum. So once it reaches the sarcoplasmic reticulum, the sarcoplasmic reticulum will release, okay, calcium ions. So the release of calcium ions will take place in response to the action potential that comes through this axon of the motor neuron. So in response to that, so we see the release of calcium ions from the endoplasmic reticulum. Now these calcium ions which are released now will go and bind to, okay, the TNC. So I told you that there is troponin. Okay, troponin C, in short, TNC, and this has a strong affinity to calcium ions. I said. So when calcium ions are released, those calcium ions will go and bind to, okay, it binds to this right, troponin C, and binding of that calcium ions to troponin C will result in, okay, opening of active sites. So troponin C plus calcium ions will unmasking result in unmasking of active sites on actin filaments. Unmasking of active sites on actin filaments. So here, okay, when the action potentials finally reach the sarcoplasmic reticulum, when when they reach the sarcoplasmic reticulum, the sarcoplasmic reticulum releases the calcium ions. Those calcium ions go and bind with the troponin C. So binding of calcium ions with the troponin C will result in unmasking the active sites on active filaments. So this will result in the binding of myosin head with this actin, with the, I mean, active site of the actin filaments. So that we'll see even the animation also. I'll show you the animation. Okay, so first we'll finish off the cycle, how actually it takes place. So you can see in this diagram, okay, so the myosin binding sites blocked by tropomyosin. So here these small holes, what you see are the active sites on the G actins of right actin filaments, and this is covered by, okay, this protein, so which you see rope like, and that is the tropomyosin. And these okay, the violet balls are the troponin complex, and one of that is TNC, which has affinity to the calcium ions. So when the calcium comes and binds, so you can see the calcium, it finally comes and binds to this troponin C. So it binds to troponin C, and because of that, okay, you can see the troponin unmasks the active sites. Now in this case you can see very clearly these active sites on the actin filaments. Now when these are present, the myosin heads will come and bind to this, and that initiates the, okay, the muscle contraction. So we'll see that cycle now. So here is a picture where I show the stages in cross bridge formation. So here whatever I'm showing are stages in cross bridge formation. Stages in cross bridge formation. Okay, right. So here you can see, so once the calcium ions bind with the troponin, when binds with the troponin C, so this is the starting point. So this is the, okay, first stage. Here the myosin head is bound to ATP, okay, and is in its low energy configuration, and you can see already the actin filaments are showing the active sites. So here when I told you that the TNC, when it binds with the, okay, calcium ions, okay, so it results in unmasking of active sites on actin filaments by tropomyosin. Okay, so once that is finished, you will see here the active sites are exposed, and now the ATP will bind with the myosin head. So we have seen in the previous okay, sessions that the myosin head will have, okay, two points where one it attaches to the actin and another one, so this is the ATP binding site. So this is the ATP binding site, and this point is actin binding site. Actin binding site. Okay, so here you can see already the ATP energy it binds. Okay, and now you can see the myosin head. Okay, so that has now energy, and it's trying to make a cross bridge. So here already it has taken up the energy, and now this myosin head, so you can see here this myosin head, it has come in contact with the active site of the actin filament, and that resulted in the formation of the cross bridge. Okay, so once that is finished, okay, so these ADP plus PI will be released out. Okay, so that is right, and what happens, there will be a bend. So here it is, pushing this actin filament in this direction. So already they have shown here the arrow mark, so pushing in this direction. Once that is finished, so this one will come back to this, okay, a normal state. It unbinds and again takes up one more ATP molecule, and again binds, and the same process continues. Okay, this results in pushing and pushing. Of course that there is a limit. Okay, so here where the H zone almost it becomes 0. Okay, so that I will show you now in an animation, so how it all happens. So this is how it actually, here one small picture they have shown you. Okay, so on either sides this is the actin filaments, and these are the myosin heads, and these myosin heads making in contact, and then okay, pull these actin filaments over each other, right, so thereby reducing the length of the sarcomere. So that is what we call the muscle contraction. Now I'll show you an animation on that. So here is the animation, so I would like to show you. So you can see this is the T tubule. So there is a T tubule here, and on either side you have endoplasmic reticulum, and this is of course inside the sarcoplasm you find the myosin with the heads, and these are the actin filaments, and this is the the Z line. From this Z line to this Z line is what we call so a sarcomere. So we'll see here the sarcomere, how it contracts. So this is the action potential from the neuromuscular junction, reaching onto the surface of the sarcolemma and passing into the T tubule. So there it triggers the endoplasmic reticulum, that is the sarcoplasmic reticulum, which releases calcium ions. So you can see here the calcium ions are released, and these calcium ions now go and bind to the troponin. So one subunit of that troponin is troponin C, that has affinity. So you can see here the white colored one is tropomyosin, the two F actin filaments. So this is the troponin, and this is, so you can see here the calcium ion binds to the troponin. That results in unmasking of active sites. You can see once this happens, these are the myosin heads, with the short arm. So that comes and binds and results in the formation of cross bridge, and you can see how they are bending by using the ATP energy. So this is how the contraction, the stages in the contraction, stages in the cross bridge formation. So we'll see one more animation on this. So right here is another animation. You can see in this animation the myosin filaments and the actin filaments, and you can see the Z line to the Z line is in fact a sarcomere what you have. Okay, and here is a region where the actin filaments are not present, is a head zone, and where, okay, the till the end of this myosin filaments is the A band. Okay, so let's see. Of course, the I band, at the center of the I band is the Z line. So when there is a contraction of these muscles, I mean, okay, let's see. Can see in this case all the different zones, and see how the myosin heads are moving and pulling the actin filaments towards each other, and finally disappearing the head zone. So completely the head zone disappeared, but the A band length remained same, whereas the I band length decreased. So I told you that the head zone decreases or completely disappears. Okay, so the A band remains unchanged, H zone disappears, and even I band, its okay length also decreases. So this is how, okay, in a fully contracted muscle you will be seeing. So this is how the contraction of the muscle takes place. So that's all about the sliding filament theory of muscle contraction. So now we'll see if there is a repeated activation of the muscles or contractions of the muscles, okay, it will require more amount of oxygen, and if this more amount of oxygen is not available, so if the muscles are in a continuous stress, then definitely it results in deficit of oxygen, and this results in the anaerobic breakdown of glycogen. So it results in anaerobic breakdown of glycogen. So this happens, okay, when the muscles are repeatedly activated. So when there is a repeated activation of muscles, the repeated activation of muscles will result in anaerobic breakdown of glycogen, and this results in, okay, production of lactic acid. It results in the production of lactic, right? And this formation of lactic acid in the muscles will actually result in muscle fatigue. So it results in a muscle fatigue. So it means it becomes tired. So of course, once the contraction of the muscles or its stress is reduced, the lactic acid will be removed, okay, and the muscles will come back to a normal state from the fatigue condition. So now here, okay, in some muscles there is a red colored oxygen storing pigment. So there is a red colored oxygen storing pigment. So the red colored oxygen storing pigment is what we call the myoglobin. So this myoglobin, uh, okay, of course, it's a high content in some muscles will give it the red appearance. So if there is myoglobin, and if the myoglobin is more in quantity, more in quantity, so it will give, okay, the reddish appearance or reddish color to the muscle. So reddish, okay, color to the muscle, right? And these muscles are what we call them the red fibers. So as this they appear in the red color, they are called the red fibers. They're called red fibers, and these red fibers have a lot of mitochondria. They contain, okay, okay, a lot of mitochondria they have, so that they provide energy, a continuous energy. And moreover, okay, because this myoglobin stores oxygen, so that oxygen is useful in breaking down, okay, the glucose in, okay, and then undergoing the cellular respiration, and this mitochondria will result in the finally the production of ATP. So the mitochondria results in production of ATP. So it produces ATP. Okay. Then there are, okay, other muscles where this myoglobin. So here the quantity of that myoglobin is very less. Okay, fine. Of course, here one more point. This the red fibers. So the red fibers are also called the aerobic muscles, because they store, okay, oxygen, and they undergoing the fatigue will be very less. Now coming to another type of muscles whom we call white fibers. So these white fibers will have, okay, very less quantity of myoglobin. So they have very less quantity of myoglobin. It has less quantity of myoglobin. Okay, and because of that, these white fibers, or you can also say they appear pale in color. So pale in color or whitish in color. So they appear pale in color or uh, whitish in color. So they appear. Right. Now in this, okay, the white fibers, in these white fibers, mitochondria are less, so hence generation of ATP will be also less. So mitochondria are less. Okay, and uh, and one more point is that in these white fibers, okay, even though mitochondria are less, mm, endoplasmic reticulum will be more, so that the endoplasmic reticulum in the muscles, what we call the sarcoplasmic reticulum. So here the white fibers have high content of high content of sarcoplasmic reticulum. You can call this in short as SR, the sarcoplasmic reticulum. And here, as the mitochondria are less, and even the quantity of oxygen storing myoglobin is also less. Okay, so these are the muscles that can undergo very quickly the muscle fatigue. So they can, okay, quickly undergo muscle fatigue. Right, and these will depend, okay, on anaerobic process for energy. So these white fibers depend on anaerobic process for energy. So this is the differences between the red fibers and the white fibers. So here these I discussed about the red fibers. Okay, so they have more content of this myoglobin, whereas the white fibers have very less quantity of myoglobin. Okay, and these red fibers produce more amount of energy, and undergoing muscle fatigue is very less. So this is all about the muscles, and in the next session we shall see about the skeletal system. Okay.